Hello and welcome to our presentation on electron microscopy. In this presentation, we will be discussing the theory behind electron microscopy, providing information about the several different types of electron microscopy, and going into further detail on the basis behind transmission electron microscopy and scanning electron microscopy. We will also examine the kinds of results obtained from electron microscopy and how this data is used to further our understanding of structures, as well as discussing the various advantages and disadvantages of using this particular technique. Firstly, there are many different types of electron microscope, as you can see listed here. In this presentation, we will be focusing on transmission electron microscopy and scanning electron microscopy, as these are the most common and universally used types. Here you can see examples of pictures captured with both transmission and scanning electron microscopes, as well as a visual demonstration of the development of electron microscopy over time. Let's talk about the physical basis behind this analytical technique. Electron microscopes use a beam of electrons that are shot towards a sample, and this is regarded as a form of wave motion due to the electron's quantum behaviour, which means that it acts as both a wave and a particle. Louis de Broglie first derived the formula in order to calculate the wavelength of an electron in the 1920s. This is that the wavelength of an electron is Planck's constant, multiplied by the speed of light in a vacuum, divided by the square root of the charge of a single electron, multiplied by the electric potential, which is then multiplied by 2, times the mass of a single electron, times the speed of light in a vacuum squared, plus the mass of a single electron, times the electric potential. You can see that the vast majority of these figures are physical constants, with the only variable being the electric potential. This means that the wavelength of an electron is determined by its electric potential, and a higher electric potential like that which is used in an electron microscope leads to a very small wavelength. This is advantageous, as smaller wavelengths lead to overall higher resolutions when compared to a simple optical microscope. As we can see on the right, the images given from an electron microscope, in particular a scanning electron microscope, have a far greater resolution than the optical microscope. It is easier to pick out intramolecular structures with the SEM, and another advantage of the SEM specifically is that it produces a 3D image, making it easier to ascertain the dimensions of your sample. Now, let's compare the differences between the TEM and the SEM. Whilst both instruments rely on the electrons being fired from an electron gun to illuminate the sample, in a TEM the electrons pass through the sample, and in an SEM electrons take advantage of a process called electron backscattering in order to form an image. Let's first look more closely at TEM. Firstly, a beam of electrons is produced by an electron gun, and this beam is focused by a condenser. An image producing system then focuses the image, and a complex image recording system, which usually involves a fluorescent screen and a digital camera, converts the image into something we can see and understand. TEM usually has a very high resolution, but it can only form 2D images. Now let's look at the SEM, where again the scan begins by a beam of electrons being fired and a condenser focusing the beam on the sample. When the electrons hit the sample, higher energy electrons are backscattered, or they fire right off the sample, and these have their own detector. Lower energy electrons are detected by a secondary detector, and these form an image. The SEM generally has a lower resolution than the TEM, but it is advantageous in the fact that it can form a 3D image.
We will now take a look into how electron microscopy actually works. The microscope contains 3 integrated sources of radiation. These are electron beams, visible light beams, and X-ray beams. The electron beams are produced by a heated tungsten cathode and an accelerated anode. The 2 electromagnetic lenses allow the beam to focus on the surface sample. The diameter of each of the beams is between 0.1 and 1 mu. The microscope has a vacuum system that provides pressure of less than 10 to the minus 5 torr. When it comes to scanning electron microscopes, the electron beams are accelerated and focused on a sample using lenses. They will interact with atoms at different depths to produce different signals. These signals include secondary electrons, which are low-energy electrons that can be ejected from the valence or conduction bands of an atom through inelastic scattering from the beam of electrons. The secondary electrons are detected and the number of electrons are determined by the variations of the surface sample. The electron beams will also ionize the atoms present and make them emit X-rays. Characteristic X-rays are emitted when the electron beam interacts with the sample and removes an inner shell electron. This results in a higher energy electron filling the shell and releasing energy. The electron beam absorbed by the sample is detected and used to create an image of the sample distribution. The final image displays a map of the intensity of the signal emitted, leaving you with a clear and detailed illustration of the surface of the sample. We will now take a look at the imaging that electron microscopes produce. As you can see in Figure 6, the analytical technique produces a black and white three-dimensional image. The 3D effect is down to the depth of the field, and since there is minimal penetration of the electron beam into the sample, only the surface of the sample is shown. These black and white images can be artificially coloured using an online software to help make the imaging more lifelike. This is done by allocating a colour to each shade of grey, black and white in the image, as illustrated in both figures 8 and 9. Now we will take a look at the advantages to electron microscopy and how it is an extremely useful analytical technique that allows us to look at both the physical and chemical nature of the surface of a sample. A huge advantage to the electron microscope is that it has many important applications throughout chemistry, biology and medicine. Some of these include the ability to study the microstructures of ancient ceramics, the investigation of grain boundaries in alloys, the measurement of diffusion rates in semiconductors, the study of active sites in heterogeneous catalysts, and for the determination of occluded species in crystals. Another advantage to electron microscopy is that it has extremely powerful resolution. This results in better imaging. They have a resolution power that ranges up to 0.2 nanometers. This is 1,000 times higher than the standard light microscope. Since the resolution is considerably higher than the light microscope, it allows us to see detailed structures that aren't visible to a light microscope. However, like most things, electron microscopy isn't only advantageous, it also has its limitations. One of the biggest limitations to this analytical technique is being the extremely high cost to both construct and maintain the microscope to a high standard. With the average cost of an entry-level scanning electron microscope costing between $50,000 and $200,000, and an entry-level transmission electron microscope costing between $100,000 and $1 million, they're a huge investment that require constant maintenance. Not only are they extremely expensive, they are also overly sensitive, bulky pieces of equipment. This takes up a lot of space in a lab, and magnetic fields and vibrations caused by other lab equipment can potentially interfere with their operation. Since electron microscopy is used under a vacuum system, samples taken cannot be used to analyse living specimens. Another issue that occurs when using electron microscopy is when studying non-conductive materials, as before they can be analysed, they must be coated in a conductive layer. Me and Lee would like to say thank you for watching our presentation.